Okay, we're going to get started. So hi everyone, I'm Jim Lodge. I'm with the foundation. Just quickly, I'm going to go over a little logistics for the room. I think most people found that we have coffee, and please help yourselves. If anyone needs the restrooms, there's keys at the front desk. Make sure you get the right one for your sex. We have a webinar going as well as the meeting here. So Matt has a great turnout here, probably one of the largest we've had, which is great. So there's about 40 of us here in the room and another 30 or so on the web. So the logistics for the web folks are they can hear, hopefully the mic's up here, they can hear relatively well. They can see the slides well, but they can't speak to us. So at the end of the meeting, when we're going to open up for questions, they'll have an opportunity to type in and answer questions. So the title of Matt's talk, Restoring Oysters Sustainably Requires Good Genes, Genomic Evidence from Delaware Bay and Hudson River. Matt is familiar to me and to a lot of us here at the foundation. We've worked with Matt over the last, I guess, 2 or 3 years now with projects primarily at Soundview Park, but others around the harbor. And so this is a great opportunity, and we're really looking forward to hearing what he has to say. On a sort of personal note, I talked to Matt a lot about the genomic stuff. I'm really amazed at what it can offer, but usually about halfway through the conversation, I can't really understand what he's talking about anymore. So what I'm hoping is that sort of with this crowd and maybe the third time I've heard a similar talk, that I'll come away with sort of the requisite knowledge to understand for the next conversation. But again, welcome Matt. Matt, I'll give you a little CV history. Matt did his undergrad in Maine. Then he moved to Alaska and did an MS in zoology. Then he moved to Georgia and did a PhD in genetics, and then some post-grad work at Harvard. Following that, he got his first real job, I guess, in Maryland. He was in Maryland for 7 years at College Park, and then for the last, I guess, 8 years, he's been at Cornell University. So with that, thanks, Matt, for coming. Really looking forward to it. And does anyone have any questions before we get started? So that moves forward.
Great, thank you. Thanks, Jim, and thanks so much for the invitation. It's great to be here. Now, I know that most of you did not come here for the genetics. You came here because of oysters, right? And many of you probably feel the same way as Jim, that when somebody starts talking about genetics, it's like genetic diversity, blah, 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 genetic diversity, blah, blah, blah. And so, there's, it's a little understandable, in conservation genetics, we are harping on genetic diversity and talking about how genetic diversity is what allows a species, a population to respond to environmental change. And this is true, it's very strong theory, and there's plenty of application and examples in the literature. Nonetheless, it's not always very tangible. Today, I want to make it tangible for you with respect to oysters, and in particular with applications in oyster restoration. If I use a term that I didn't explain sufficiently, please stop me, raise your hand, because I'm not assuming that you are coming to this with any genetics background, so I'm trying to make it accessible to everybody here. All right, so let's make sure everybody's on the same footing here in terms of the biology of this beast. Oysters are just an amazing species. They're broadly distributed from Canada to Mexico, so within that species range, they're dealing with a lot of different environments. They, like many marine invertebrates, have very high fecundities, so what we're looking at here with life history, we've got adult females and males. Females will be spawning synchronously with the males and their sperm, spawning millions of eggs. So very high fecundity. Fertilization occurs in the water column and then quite rapidly develops into a veliger stage larva that is floating around in the water column for 2 to 3 weeks. All right, so 2 to 3 weeks of dispersal with the tides coming in and out allows for a lot of diffusion, a lot of advection in the water currents, and really no good reason why an oyster is going to come home and sit down next to mom. Okay, so when we have an oyster reef restoration project and we're putting out oysters and we want them to be reproducing and producing larvae, those larvae are going to go everywhere, and very few of them are probably going to come back to the reef that we've worked so hard on. So to finish the life cycle here, at a point where they become competent to settle, they get this little foot, they settle down, and we call them spat at that stage, juvenile oysters. And then they develop over in these temperate areas like New York, in 2 to 3 years become reproductive adults. Okay, so one of the things I'll be talking a lot about today in terms of oyster biology is phenotypic plasticity. This is the idea that a particular genotype, okay, so the alleles, the genetic variation contributed by the sperm and the egg to a zygote creates a unique genotype, okay? It's ephemeral. It will never happen again. It's never happened before. There's so much variation contributed by these two gametes that come together and act together in a diploid zygote, right? We always have a chromosome from mom and one from dad for each type of chromosome. That genetic variation is unique to that individual. By producing millions of eggs and getting fertilized by different sperm, each female is putting out a whole lot of genotypic diversity. And with probably a diverse array of phenotypic capabilities among those genotypically diverse zygotes. So this plasticity, however, refers not to the diversity we get from genotypic diversity, but for any one of those genotypes, one of the beautiful adaptations of oysters is that they're phenotypically plastic. Depending upon the environment that they find themselves in, they can osmoregulate at low salinity, they can control their cell volume and in relation to salinity at high salinity down near the ocean. They modify their shell morphology depending upon where they're growing, on a piling or in an oyster reef. They can grow in the intertidal and survive the hot sun versus subtidal. Euryhaline refers to this idea that they can exist, they can thrive across from nearly freshwater up high in estuaries. This is Delaware Bay, but right now I'm using it as sort of your canonical estuary, and down to oceanic salinities. All right, down here near the ocean, they're going to suffer ecological constraints, a lot more predation. And up here, freshwater, they don't do very well. So on the right-hand side, what I'm showing here is a recent effort to integrate across a lot of literature, speaking to the salinities shown across here that make oysters happy, okay, that allow for strong performance. And so this is oceanic type of salinities, this is freshwater at 0 salinity, and this is referred to as an oyster suitability index. You can see that it's the moderate salinities where oysters have the fastest growth rate and it's most suitable, but that you can find oysters in most estuaries across the full range of salinities. So that's an example of plasticity that we've looked at in my lab and that I'll describe for you today. So one of the main questions that I want to address today is how much does oyster population fitness, that is the fitness of a population within an estuary, depend upon this adaptation allowing for plasticity, in this case the physiological plasticity of dealing with changing salinities as the tide goes in and out, or when a larva settles way upstream at low salinity or way downstream near the ocean. How much of that is due to plasticity, or are there evolutionary mechanisms, either short-term or long-term, that depend upon genetic diversity, that confer population fitness, that confer the ability of the population to grow and maintain abundant oysters? I also want to address, because they're related questions, So if there are evolutionary mechanisms acting on the capabilities of oysters across this environmental gradient, natural selection, Darwinian natural selection, often acts across multiple generations to differentiate, functionally differentiate populations within local environments, right? Local adaptation. Gene flow, the connectivity of genes by larvae moving between populations will tend to homogenize genetic variation, okay, and will work against local adaptation. So there's a balance between how much local adaptation you'd expect versus how much gene flow connectivity is happening. And so I want to use genomic data to ask, is larval dispersal within estuaries, in particular in the Hudson River, limited between upper estuary and lower estuary? And in particular, we might expect it to be limited in the upstream direction, because larvae will, by and large, move downstream in an estuary. Because if this is true, it would allow for some local adaptation for those upstream low salinity populations. And finally, because in recruitment-limited estuaries, right, where there's just not that much living broodstock available, there's not very dependable recruitment of spat each generation, we're typically for restoration using hatcheries to produce cohorts that we plant on restored reefs to try and kickstart things. And so it's important to ask, what are the genetic effects of that? And so I'll show you some data that speak to the degree of genetic bottlenecking that occurs in hatchery production. All right, so the empirical systems that I'm going to use to talk about and address each of these questions is, first of all, the Delaware Bay system, looking at plasticity in terms of gene expression. Okay, now you can you have a lot of gene expression plasticity, right? Every time, well, right now you might need a little more adrenaline, right, to set up to change the genes that are allowing you to stay alert, right? Even though I'm talking about blah 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 genetic diversity. So that's plasticity. That's it's a physiological plasticity that in mammals comes from the endocrine system. Here we're talking about the plasticity of gene expression relating to osmoregulation and the ambient salinity. Oysters are osmoconformers, so their hemolymph, their bloodstream, is isotonic. It's the same salinity as their surrounding water, but their cells have to maintain proper volume, and so they have to work pretty hard physiologically to keep that osmoregulatory control on cell volume. Then we'll move to the Hudson River to look at population structure, and finally we'll compare hatchery-produced oysters versus wild oysters, again from the Hudson River estuary. All right, so why do we want to restore oysters? I think it's important to back up and reconsider what our objectives are and our motivations are with oyster restoration. In New York City, to me, it's very compelling that there's a legacy of oysters in this estuary, and in New York City in particular. And Kurlansky's recent book describing the history of oysters and the Big Oyster, as it's called, right, before it was ever called the Big Apple, I think. And so just the idea that we had oysters, commercially vibrant industry, as well as an estuary that benefited from oysters only 100, 150 years ago, means that it's not long past, and it'd be great to get it back. But in addition, there's a lot of very tangible and important ecosystem services that this species can provide when there are healthy populations in an estuary. So what this term ecosystem services mean is that for a species that has a lot of ecosystem effects, we call them a keystone species. And in particular with oysters, they're sometimes referred to as an ecosystem engineer, because an oyster reef, the three-dimensional structure of an oyster reef, provides habitat for many other dozens, hundreds of other species. Incidentally, I'm using an illustration here from the Billion Oyster Project Oyster Gardening Manual, with Pete Malinowski kindly provided to me. And it's a beautiful manual. And so we can see the habitat provisioning here. They're doing a lot of, the way they eat is filtering the water, right? And so they're filtering several liters per hour per oyster. And so overall, an oyster reef can pretty much clarify the water if it's a vibrant reef. And that allows for a lot more sunlight penetration to seagrasses and other aquatic vegetation. And then finally, reefs can provide some protection to the coastline by attenuating wave strength. So I also wanted to just, you all, many of you are involved in oyster restoration in the area, so you'll know all this. But I think I've only been in the New York area for about 8 years now, and it's impressive to me how much interest there is in oyster restoration. I just wanted to list the projects, not go into any detail, but the Billion Oyster Project I've already mentioned. There's Pete with the students at Harbor School. Oyster Restoration Research Project and the Hudson River Foundation, and here's some baykeepers volunteers at Soundview. The Tappan Zee Bridge Reconstruction Project has some money devoted to mitigation for oyster disturbance, and so I've been working with others and ATRF on a mitigation strategy there. And then there's several other either current or planned projects, some of them very large, like the Rebuild by Design ambition of having fringing oyster reefs all along Staten Island. So in many cases, the goal behind these oyster restoration projects, well, of course, all of them in these New York and New Jersey closed waters, is not to produce consumable oysters. Okay, these are all closed waters. They're healthy enough waters now, thanks to the Clean Water Act, that oysters can survive and perhaps even thrive. But they're not going to produce oysters that you want to eat. Okay, and so the point of restoration is not for harvest. Rather, it's for the ecosystem services. And in many cases, these restoration projects are small scale. They're pilot scale to sort of research how to best accomplish restoration goals, to understand the oyster biology a little better. These tend to be high maintenance projects with local benefits. Ultimately, what we want to be able to do, though, is enhance the population and build it up to a point where it's self-sustaining. Okay, that's not going to happen at the local single reef level. That's going to happen when we can generate a full interconnected metapopulation, that is a series of populations that are sources and sinks for larval exchange. And so we want to be able to jumpstart this process, and that's going to involve stock enhancement using hatcheries. And so I think the goal, the long-term goal, needs to be rebuilding these self-sustaining reefs. To do that, we don't want to just put oysters out so that they grow a little bit and filter some water. We want to put them out there so they reproduce, and that their larvae will survive and settle down on local reefs and reproduce. All right, so then a little more about the plasticity with respect to osmoregulation. This is the figure I showed you before, so showing you the range of mean salinities that we can expect the average oyster to be able to deal with. And here it's plotted in terms of the minimum annual salinity. So the minimum, often during the spring when there's a lot of fresh water coming down the estuary, that the oysters will survive is between 2 and 10 parts per thousand, and down below 5 parts per thousand, you're looking at pretty low survivorship. And so it was with some amazement that when Jim Lodge and Mark Rees at EPA worked with a sonde of mine, a data recording device of mine at 2 meters depth up near the Tappan Zee Bridge, that we recorded in March to July of 2014 this trace of salinities. Okay, so in the spring, in March, there was some strong precipitation events, and several days on end where there was almost zero salinity. We're interested in this particular spot because there's it's pretty rich with fairly sizable adult oysters. Okay, so this is really nice to see oysters living up there that high in the Hudson River, and it's this capability that amazes me. So is that capable? Is any average oyster capable of that? Right, which would be suggested by phenotypic plasticity. Or are there only certain genotypes that are capable of this? And do we have a marginal population there that is evolutionarily locally adapted to those conditions? This makes a big difference in terms of how you're going to choose broodstock to propagate a cohort in a hatchery for planting in a restoration reef here or there. All right, enough of the introduction. Let's move to Delaware Bay so I can tell you about work of a previous graduate student, Laura Eierman, who collected oysters, adult oysters, from a low salinity reef at around 10 parts per thousand salinity in Delaware Bay and a high salinity reef. She brought these adults to the hatchery where she put them in tanks for 9 weeks. Okay, so from the low salinity reef, she put them at what felt like home, 10 parts per thousand salinity, and she stressed them out by putting them up at 30 parts per thousand salinity. Same thing for the high salinity guys. Okay, so here I call it moderate salinity because it's about 25 parts per thousand out there. So here they feel at home at 30 parts per thousand, and then there was 6 other individuals down here stressed out at 10 parts per thousand. 9 weeks of acclimation. It's like, all right, just gotta get used to it, right? So that's sometimes what phenotypic plasticity requires some time to physiologically adjust. And then let's look at what the genes are transcribing, what the genes are producing in terms of the transcript, the RNA that goes to produce a protein for all the genes that are active in the gill tissue that we sampled from these adults. So I'm using a technique called RNA-seq to just sequence all that material from each one of these individuals, and using EdgeR to actually test for patterns of differential expression. That is, the low salinity oysters express a particular gene more than the high salinity oysters. And how different was the expression for low salinity oysters when they're at 10 versus 30 parts per thousand. Okay, so there's a few different ways that we can test this. Our general expectations, I'll show you with this cartoon, they're often depicted in terms of what's referred to as a reaction norm, which is simply a fancy term to talk about the phenotypes that you would expect given some plasticity across different environments. Okay, so here I'm showing you 2 environments. Let's say it's low salinity and high salinity. And our phenotype in this case is the amount of gene expression. Okay, so for each gene in the genome, it's getting expressed at a certain time or place, depending upon the environment, and it might be expressed by making a lot of protein or by making just a little protein. And this is presumably something that's, in many cases, adaptive. Okay. So what we would expect in the kind of experimental design that I described for Delaware Bay, if we think of genotype 1 as being the low salinity reef and genotype 2 being the high salinity reef, we'll refer to them as genotypes as if these are 2 distinct, genetically distinct populations, but that's actually what we're testing. And under the genetic scenario, the one hypothesis is that the genes show no plasticity, no change in the expression level for any given genotype when you put them in different environments. But we see a very big difference between the genotypes in their gene expression level. Okay, so that's a population effect or a genetic effect. The 2 populations might do very similar things, and both show a similar trend in terms of phenotypic plasticity. Okay, in this case, both showing higher gene expression in the second environment. But there's no population effect here, right? They're both acting similarly. And then if both are happening, there's an interaction effect, a genotype by environment interaction, so that they're both showing a pattern of plasticity, but it's a different pattern. Okay, so they've adapted to have a different plastic response when the treatments are these 2 different environments. Okay, so that's a genotype by environment response. So what did we find when we analyzed all the genes that were expressed in these tanks, in these oysters? Let's look first at, so in this Venn diagram, what we've got here are, we're statistically testing for differential expression that was consistent with a plasticity effect here, a population or genotype effect here, and a genotype by environment effect here. Okay, so our 2 factors and their interaction. And depending upon the particularities of statistical power, sometimes genes are in the middle and statistically significant for 2 or more treatments. But if we look at the numbers that refer to genes that were significant for only one factor or just the interaction, we see initially that this plasticity pattern, which we expect to, we know oysters are plastic, so that seems to be the genomically most pervasive pattern. Okay, many genes were responding, thousands of genes were responding differently at low versus high salinity. There were very few genes that showed no plasticity, but a difference between the 2 reefs, the low salinity source and the high salinity source. Okay. But it was also, this genotype by environment pattern was also genomically pervasive. That there were thousands of genes that showed a different pattern of plasticity, depending upon whether, that is, different pattern of plasticity going to low or high salinity tank, depending upon whether you got that oyster from up estuary or from down estuary. Well, so I can't say from these data, these are phenotypic data, right? Gene expression is a phenotype. It's going to depend upon the genotype, but these are phenotypic data. So I can't say from this that this is necessarily indicative of genetic differences between these reefs. Okay, we only acclimatize these oysters for 9 weeks, and the ideal method, if you're working with Drosophila or something else, is to actually breed them, reproduce, and do the test on their offspring, or then maybe a couple generations out on their offspring, because there are mechanisms like epigenetic mechanisms that can have legacy effects. Okay, so far I've only shown you patterns of plasticity differ. That is, the functional response of oysters to different salinities differs depending upon where they grew up. But I wanted to get a little further, and the beauty of this approaching gene expression measures this way through sequence data is that we can actually look at the DNA sequences. Before we go there, I want to just make the point. Gene expression plasticity is a phenotype. It's not the whole story. I'm interested in understanding whether there are evolutionary mechanisms at work here. And so to do that, I need to look at things that are changing allele frequencies at the genetic level. Allele being simply a genetic variant. And if genetic variants are at different frequencies in the 2 reefs, that would indicate, despite the fact that larvae are homogenizing genetic variation every generation, that would indicate that there's strong selection after larval settlement, right, during the growth from spat to adults. We know there's a lot of mortality then. We don't know yet whether there's strong selection. So every one of those messenger RNA transcripts that's produced at a gene that we're measuring the number of those in order to talk about the degree of gene expression, every one of those has a DNA sequence or an RNA sequence. And so I'm showing you some in this cartoon, several transcripts with a sequence of nucleotides G, A, T, and C. And the type of variation that we can score, that I've scored with these genomic data, are referred to as SNPs, single nucleotide polymorphisms. And all that means is that at a particular position, there's 2 different nucleotides that occur. Okay, so that's a type of genetic variation. If several of those occur within the sequencing read that we have to look at, it defines a haplotype. Okay, and so this haplotype is the sequence of these 3 variants, A, T, and C. But there's other distinct haplotypes like A, C, G, or G, T, C, because of these 3 polymorphisms and the way they line up. Okay, so we can look at so the results I'm showing you today. I've analyzed it both in terms of SNPs and in some cases in terms of haplotype variation. Any particular SNP, any one SNP, and we're only looking at 2 variants that are segregating in the population. For haplotypes now, we're looking at many possible variant sequences that are segregating in the population. So that's sort of the difference of looking at it versus with SNPs versus haplotypes. All right, so now going down to the genetic level, if we think of these genetic variants and their frequencies in the two reefs, FST is a statistic that describes the difference in the degree of difference in the frequency of those genetic variants between two samples. Okay, it goes between 0 and 1. 1 would be fixed differences for different variants. Okay, so they don't share any variation, whereas 0 would be the same allele frequency in both populations. And I'm using a particular quantitative measure that is unbiased, and so it sometimes takes negative values. You can just think of those as 0. So across the almost 80,000 different genetic loci, genetic variants that we're looking at, the overall average FST in our data was 0.0025, pretty close to 0. Okay, and this is what you would expect for a high gene flow species where larvae are really mixing it up every generation. However, the distribution of FST had this long tail, okay, and that was of interest to us. The top 1% of that distribution included almost 800 SNPs with FSTs between 0.3 and 0.73. Okay, so some pretty high differentiation at this small subset of genetic variants. So we wanted to use these genetic variants, and in particular these what we refer to as outliers. All right. Our hypothesis is that most of the genetic variation is not responding to environmental gradients. Okay, and so sometimes that's referred to as evolutionarily neutral variation. Okay, it's governed. The frequencies are governed by genetic drift, not by selection gradients. Perhaps this tail in the distribution is telling us about loci that are responding to environmental gradients, and in this particular case, the gradient is a salinity gradient. So with strong selection, what should we see? We would expect to see, let's say at the low salinity reef, lots of larvae settle down, but it's only a few, but it's only those genotypes that settle down that have a particular genetic variant, say a particular sequence or allele, that can make it at low salinity. Okay, so that very strong differential viability selection is going to lead to what? At that gene, we're going to see reduced genetic variation at that gene compared to other genes, other neutral genes. And indeed, we saw for the low salinity reef, measuring genetic diversity in terms of, this is a statistical measure of nucleotide diversity, we saw significantly lower nucleotide diversity in these outlier genes, these 700 outlier genes, at the low salinity reef compared to the high salinity reef. Okay, indicating perhaps there was stronger selection at the low salinity reef. It's a more marginal environment, kind of makes sense. The other expectation is that if there's strong selection to a single sequence or a single haplotype, it's going to make the variation associated with each SNP, each single nucleotide polymorphism, show correlations in the frequencies along that sequence. Okay, this is referred to as linkage disequilibrium. This is getting a little technical, I know. But the point is we had two expectations, and looking at the plot of those two expectations together, now for these outlier loci, and here we've got average nucleotide diversity measured at a per exon scale. Every gene is sort of organized in terms of exons, and so these data are analyzed at the level of exons. And we see that some of them showed average levels of nucleotide diversity, but there was quite a few that showed these very low levels of nucleotide diversity. I already showed you that. This is for the low salinity reef. In addition, those with particularly low nucleotide diversity also showed these stronger correlations in terms of the SNPs along that sequence. Okay, so we saw a significant correlation here, which is indicative, we think, of strong selection on this low salinity reef. All right, so what does this mean? Why is this guy jabbering on about selection? If every generation involves this 2-week larval dispersal, homogenizing all the genetics, and now, later we'll talk about, okay, maybe it's not complete homogenization, but let's just assume the idealized model of complete homogenization. Then where would these genetic patterns come from, right? what sort of selection can lead to as differences in nucleotide diversity and differences in gene expression? Well, in fact, if selection is strong enough and fecundity is high enough, so there's zillions of spat settling out, but only a small subset of that growing up to be adults, then either that mortality is all stochastic, or there's likely to be strong selection in terms of the microenvironment that the oysters are living in. Okay, so I think this is a process that's going on every generation, recurrent selection generating locally adapted genotypes across the environmental heterogeneities, even at this very small scale within single estuaries. And then it all gets mixed up again in the larval pool next generation. However, to the degree that there is a demographic, some measure of demographic isolation within an estuary, say for the upper estuary populations, it's possible that in addition to this within generation process, there's a more multi-generational, classical Darwinian pattern of local adaptation also contributing to the genetic differences between low salinity and high salinity reefs. So that's what we wanted to test for, and we moved, so that's what that says. That kind of Darwinian adaptation is only likely if there's some gene flow limitation, if there's limits to the connectivity, larval connectivity between, say, upper estuary and lower estuary. Biophysical larval tracking models in Delaware Bay and Chesapeake Bay suggest that there's a fairly strong asymmetry, that larvae mostly move downstream. So this may set up some gene flow limitation moving in the other direction. And we're currently doing some simulations to test these ideas about the degree of within versus multi-generational selection that's contributing to the differences that we see. So I want to move now to the Hudson-Raritan estuary and samples that we did with the benefit of New York Sea Grant funding. This is work where Yuka Katsumi has done a lot of the hard work here in terms of the lab work and bioinformatics. Our collections were from both 2012 and 2013. I had the benefit of a lot of help, both in the field and getting our field sites established and monitored, but also Jeff Levinson provided several of these Upper Hudson samples, and New York City Parks helped me get out to Prowles Island. All right, so I'm going to jump straight to a result, initially with microsatellites. So microsatellites are simply a kind of marker. It's a marker that's showing you some polymorphism. It's a highly diverse marker, so whereas a single nucleotide polymorphism only has 2 variants segregating, microsatellites tend to have in oysters 10, sometimes 30 different genetic variants at a particular locus. So they can be very informative. Okay, that's why geneticists use them to try to understand population processes. This is where we started before we got genomic data, and it's where all the previous oyster population genetic work had been done, was with microsatellites. Well, it's not entirely true, but a lot of it. And so using 8 markers to analyzing samples from the sites that are shown in red outline here, in addition to Fisher's Island adults, Fisher's Island being aquaculture oysters. What I'm showing you is a bar chart that is the result of an analysis, a Bayesian clustering analysis, that treats the multi-locus genotype of an individual as a unit of analysis. Okay, so so I'm an individual. I've got if we pulled out 8 microsatellite loci from me, it would show you a particular series of alleles. That's my multi-locus genotype. And so each thin vertical line here is on this bar chart is an individual, and the colors reflect the source populations that seem to be contributing variation to that multi-locus genotype in an individual. All right. One of the so you don't tell this program how many populations you have. You infer how many populations you have from the data. And with these microsatellite data, and with Fisher's Island included in the analysis, the data were most consistent with an inference of two differentiated populations. And if I took Fisher's Island out, the data were most consistent with a single undifferentiated population. Okay, as you can see here as a single color of red. One of the things that we were interested in is since Fisher's Island oysters have been used for restoration, some in the East River, do we see their influence? Were they contributing larvae that were then coming in and settling on our settlement plates? Or in the recent past, had they contributed? And it appears that maybe they'd had a little bit of contribution in some of these in Soundview. All right, so that was so microsatellite is kind of low resolution, telling us complete homogeneity. Then we went to the genomic methods. If you're none of this is detail you really need to get the punchline, but for those who are interested, it was a double digest RAD approach. Basically what this is a way of random sampling the genome when you don't have enough resources to just sequence the whole thing, okay? If you have a really large genome in the species you're interested in, you need to sample that genome, just like we need to subsample populations in order to infer population processes. So in the end, using this RAD-seq approach, we're basically looking at about 0.1% of the genome. Okay, very sparse sampling across the genome, which means we're going to sample a lot of variation that probably doesn't respond to selection gradients in the environment. But also, by chance, we're probably going to randomly sample some variation that is responding to the environment. Okay, so we expect to have both kinds of variation represented in the data, unlike with the microsatellites, where they're really they're not expected to respond to selection gradients, and they're telling us mostly about demographic history of the population. All right, so that's all technical details. All right, so what we're looking at here is initially just the wild oyster samples. This includes spat and adults from 2012 and 2013. And I've parsed the data. We had actually 24 individuals from each sample, but in order to actually stringently clean the data away, clean away any potential artifacts and biases, I mean, really it's a little silly because you, in genomics, at least in non-model genomics, we're going to great lengths to sequence and collect all this data. Mostly, we end up throwing most of it out, right, in order to filter down to very clean data that can give us patterns that we believe. So after all that filtering, we're just looking at 13 individuals per location. And again, we're using structure to describe the genetic variation. And so now you can see this is number 1, so it's the highest Hudson population, and we've got basically a latitudinal gradient here, with Prawls Island being at the far right in this plot. And so what we're seeing now, did I have, no I didn't, yeah, so after all that filtering, we're only looking at data here for 430 loci, but that's plenty of data in order to get a pretty high resolution picture of genetic differentiation within the Hudson-Raritan estuary. And what the pattern shows is that there are two source populations contributing to variation in these Hudson River oysters. I don't know what those source populations are. Okay, the data are most consistent with this fact that there are two differentiated populations contributing, but it doesn't tell me where those populations are or what they are. You can see that this gap, this spatial gap, is this break right here. And by and large, the lower Hudson populations are getting much more admixture, much more integration of genetics from this red source population, whatever it is, than in the upper Hudson. That speaks to demographic isolation, right? If larvae were completely homogenized across the system and gene flow was very, very high, there's no reason why we should see any of this genetic difference in terms of the effects of admixture. But what is this admixture?
Sorry, are you talking adult oysters, like from Fisher's Island and Soundview, that are those?
So far, there's no Fisher's Island on here.
Oh, the nursery one. I'm sorry, the one in the harbor. Which oysters are those?
Those we will look at in the next slide
Okay.
are the 2012 sample is a sample of what got placed on Soundview Reef. 2013 came direct from Pete, and so I don't know if they were involved in restoration, but they're also a Fisher Island source.
Okay.
Okay.
But the Soundview adults, then, which are not, oh, yeah, they are. Are those wild off reef?
Yes. So those were from the intertidal. Thanks for clarifying questions. So it's interesting that there's this this pattern of high admixture, very mixed genomes in these populations. Perhaps there's genetic variation in Long Island Sound, which is out here to the right of Soundview, that is contributing to this red pattern. Okay, maybe our second differentiated population is out in Long Island Sound, and that's why we see more of admixture here than we see in Prawls Island or up in the Upper Hudson. Okay. Either way, I don't really care right now about the details of where these genes are coming from. Obviously, I want to follow up to do that, but right now, the important point that it's telling us is that there is some limits to the gene flow between Upper Hudson and Lower Hudson. Otherwise, we wouldn't see this difference in the amount of admixture across the system. One possibility that I already addressed with microsatellites, but you might think, okay, well, maybe this is variation that's coming from those Fisher's Island oysters, right? So let's put the Fisher's Island oysters in there, and now you can see that instead of inferring 2 source populations here, we now have a data set where we infer that there's 3 source populations. Fisher's Island is not contributing this red admixture, but we do still see a little bit of Fisher's Island influence in the oysters, in the adult oysters that we collected from Soundview. Soundview is 13 through 16, so that's these guys, and there's a little bit of Fisher's Island blue pattern in there. Okay?
You didn't anticipate any from Soundview going up to Hastings. There wouldn't be a mix that far apart, you would imagine.
You know, we know so little about what larvae do in the water column,
Yes.
that we can have very sophisticated people developing very sophisticated models to track where larvae are going, but they're based upon assumptions of how larvae behave when they sense particular aspects of the environment, which were things that were measured in a flask in a lab, right? So are larvae going to do what they do in a flask when they're out there sensing the environment? So I don't, those models are great, but they're just hypotheses for where larvae would go. Most of the models would not predict that a larva would go from Soundview all the way up to the Upper Hudson, at least not in one generation. But over multiple generations, you can get this kind of stepwise effect that would lead to gene flow.
There's got a big gap there between the Hastings and then going down to Soundview. I mean, Soundview isn't even on there, that's right. I mean, why not sample all the places, no oysters?
Limited funding, basically. I mean, so, no, there's, we can find oysters there. It's, with the funding that I had, I wanted to get these more marginal environments, and then because this is where a lot of the restoration activity is, I wanted to have this well sampled. But no, I completely agree. It'd be nice to have that gap sampled.
Money counts.
Unfortunately. All right, so on to the third question. Does it matter that we're using hatcheries to produce oyster cohorts that are being used in restoration? And it's amazing, there's practically no data in the literature on this topic. And yet in the best practices, what Nature Conservancy will tell you, best practices for oyster restoration is to minimize genetic bottlenecks in the hatchery and use wild broodstock, local wild broodstock. But, you know, sort of like I was saying at the beginning of the talk, those are sort of general, non-tangible recommendations. Doesn't really matter. Well, it certainly matters in terms of the genetic diversity of the oysters, whether they're coming through a hatchery or not. I'm showing you genetic diversity measured in terms of the percent polymorphic loci. Okay, so among all the loci I'm able to look at genomically, only a small portion are polymorphic, and you see that in any particular species. It's just going to be a small proportion that are showing a polymorphism in the populations. The wild populations are all showing a little better than 3% polymorphic loci. And I've got two cohorts that were produced in a hatchery. First, two different samples of Fisher's Island oysters, which are showing a depressed level of genetic diversity by about a third. Interestingly, for our work, we had Greg Rivara, the Southfold hatchery in Long Island, produce oysters from wild Soundview broodstock now. So this is just a single generation of hatchery propagation from a mass spawn, and they show a diminished genetic diversity by about the same amount as Fisher's Island oysters. I'm still trying to understand why those two are the same, and so I can't tell you in any more detail why. The take-home message is that hatchery production using conventional methods will produce a fairly sizable reduction in genetic diversity for that cohort. And there are fairly simple ways of modifying how the oyster spawning and reproduction is done in the hatcheries that can lessen this bottleneck. So do we know whether that makes any difference? Is it worth a little extra trouble in the hatchery? Unfortunately, we don't. So I've shown you genetic patterns that indicate that genetic diversity is important in estuaries and the estuary populations. Oysters need that because females are sending forth lots of genotypic diversity, only a few of which are going to find a match between their genotype and the environment. We hamstring them if we put out oysters that have less genetic diversity. But what we really need are comparisons of oyster performance, growth rate, survivorship, fecundity, depending upon the hatchery protocols that were used to produce the oysters, how much of a genetic bottleneck did they experience, and also hopefully comparing them to wild oysters. Those are the data that I think are going to become convincing in terms of whether these genetic factors make a big difference. We may find that even though an aquaculture oyster that looks great on the half shell and grows very fast doesn't have the survival attributes and fecundity attributes that we need across a lot of environmental conditions compared to a slower growing crenulated wild-looking oyster. All right, so I want to leave you now with a data set that I had the benefit of seeing up in Quebec City just a few days ago at the Benthic Ecology meetings. We have the benefit of having Dominic with us today. He's from Macquarie University, and he shared this data slide that he presented. He was working on rock oysters down in Australia, and there's very few data in the literature comparing wild oysters with selectively bred oysters as carefully as Dominic did here across a gradient of environmental stress. And so what Dominic did was glue oysters down onto plates, that were either white, and so they were experiencing ambient temperatures, or gray or black. And so with black, they were going to heat up, thermal conditions that were about 10 degrees greater than ambient. And so we're looking across a thermal stress gradient here and looking at selectively bred rock oysters. They've been bred for about 20 generations to serve for aquaculture versus wild adult oysters. And survivorship, you can see here at ambient, was a little better. This is after 3 months in the summer for the wild oysters. Significantly better here, but not significant. And then again, significantly better survivorship in the wild oysters compared to the selectively bred oysters, particularly when they're super stressed out. Okay? So these are the sorts of tests we need with Virginica. All right, wrap it up now. To remind you, I'm asking about how much does oyster population fitness depend upon plasticity? It'd be great if it was just plasticity, then one oyster would be as good as another for broodstock. But I don't think it's just plasticity. They have incredible abilities with phenotypic plasticity, but I think there's very strong evolutionary mechanisms that are operating each generation. Those evolutionary mechanisms operate on standing genetic diversity, and so the population needs as much of that as it can. Is larval dispersal limited? The more it's limited, the more there's a possibility of multi-generational local adaptation. And I've shown you, at least in the Hudson River, there are some gene flow limitations. We're still trying to understand the degree of isolation there. And finally, we've documented that hatcheries even careful hatchery protocols will produce some genetic bottlenecks, okay? The question is, does it matter in terms of oyster performance? And so I'm not saying, so we have to use hatcheries. I think we can use hatcheries. We just need to find the best ways to minimize the genetic bottleneck and test the degree of performance deficiency resulting from those hatchery procedures. Let me just thank lots of folks that were involved in both the Delaware Bay and Hudson Raritan estuary studies, my funding sources, and for this talk, I appreciate the invitation and Dominic's lending of his data. Thanks very much for your attention.
Questions?
Yeah, I'm just curious, you know, we have oyster middens up in Montrose and Croton. Can you extract the DNA from the oyster middens and compare that? You know, the Flowers brothers up in Irvington, they put seed oysters in the 50s. So I'm just curious if the Tappan Zee oysters are from the Flowers brothers seed stock, or were they native oysters that could be dated back even to the woodland period? So,
You know, I'm not going to tell you that it can't be done, because as soon as I say that, somebody's going to figure out a way to do that. The hardest part is finding some tissue on those midden shells. If you can find a little dried tissue, I think we can probably get some DNA out of there and assay some of the variation using the same archaic DNA methods that have given us the Neanderthal genome. That would be fun to do.
but as far as the Flowers brothers, you know, if they put seed stock in the 50s in Irvington, you know, is there, by looking at the oyster, I'm just curious where the Tappan Zee oysters came from, if they're native oysters or they were migrants from the seed stock.
Yeah, I mean, so far I don't really have enough reference populations to be able to say anything about that, about whether the Tappan Zee oysters, Upper Hudson, came from earlier Flowers activity or whether it's wild stock.
They won't let you bring them over there. I talked to Flowers years ago, and they wouldn't let me even touch their oysters in the Hudson River. They wouldn't allow it.
Yeah. So there's a question here?
Yeah, all the, well, not all, the hatcheries around here are in fairly high salinity situations. So there are no hatcheries down at the lower salinity end. And perhaps, you know, that's why the hatchery oysters have had generations of adaptation to higher salinities and have, you know, not got, you know, genotypes that make them more suitable for the lower salinity.
You make a good point that most hatcheries are not working in low salinity waters, and most restoration is not at low salinity. Most restoration is trying to make the most of few resources, and so they're going for those moderate salinity, ideal condition locations for reef restoration and for hatchery plantings. And that's a very wise approach. However, in some cases, like the Tappan Zee Bridge, there's going to be reasons for mitigation or other reasons why we do want to help build up stocks in a more marginal environment. And in those cases, I agree, there are, one of the constraints is that most hatcheries are not set up for doing low salinity work. And my experience and experience of some hatchery managers that I know, those low salinity oysters can be pretty challenging to work with in a hatchery. They don't spawn as readily as others. So I completely agree. There are going to be constraints in using any particular broodstock that you target from different environments. So we'll move back.
I'm just wondering if you could talk a little bit more about some of the modifications to hatchery methods. I mean, in some ways it's not surprising that you have less diversity because you're probably taking, and even in the wild populations, you're probably taking from a couple areas or a few areas. So manageably, how do you kind of deal with that genetic bottleneck?
So instead of, I actually wouldn't recommend only working with broodstock from the one little microenvironment where you plan on putting out the oysters, right? If the point is to have a put out oysters that can grow, reproduce, and put out larvae that are going to have more general genotypic capabilities, the more diversity, local diversity, that you can include among the broodstock, the better. And then it's a question of getting as many of those adult oysters contributing to the cohort as possible. A mass spawn, it's well documented, tends to have skewed reproductive contributions so that only a couple of those spawning individuals are going to be the parents for most of the larvae that make it through the hatcheries. So most of the improvement can come from not exactly strict pairwise crosses, but a modified design in which you're allowing for initial fertilization more akin to pairwise, and then lumping all the larvae for the culture in a single tank. So I guess without going deeper into it, that's some of the improvements that I think could occur. Yes?
I can see how you're addressing a one-generation issue with this basically looking at the hatchery versus non-hatchery, but I mean, when you're talking about reef building, you're talking about multi-generations. And I heard, I think right in this room, I think from the Carolinas, they were looking at actual productive capacity of reef systems where you actually have selection first off on the larvae in that water column that you're releasing them into, then on the reef in that situation. So you're actually looking at a multi-generation environment, and obviously according to Darwin, the surviving oysters would produce the most offspring and they would be the most likely to settle. And actually just kind of a related question, you talked about stress, and I mean 10 and 30 are good numbers, but where are you actually hitting osmotic stress for the oyster? I would guess it would be lower than 10. So I mean, that you're obviously getting two poles, but osmotic stress would depend on osmoregulatory mechanisms, which probably has something to do with the selection issue as well.
Yeah, great questions. Let me take the last one first. Question of well, what kind of salinity actually stresses out Crasacea virginica? I think that I think our data suggests that depends upon the genotype that you're working with, and I don't want to suggest that we have a very detailed knowledge yet about the stress mechanisms of virginica and how salinity stress affects them physiologically. I think our data speak to the fact that the answer to that question will depend very much on where you get the adults that you test. And most of the literature is filled with physiological tests on adult oysters that were taken from the ideal conditions, right? We really don't know what the physiology of a low salinity oyster is because nobody's ever collected oysters from low salinity to do their studies with. So I think it's an excellent question, and I'd love to see some research focused on those genotypes. The first part of your question was...
Basically, you're looking at reef is multi-generational.
Yes, the multi-generational.
And also, I'm thinking larvae are, if you, another way to go, say, not necessarily down the road of just the hatchery, is to basically be producing larvae in the environment where you want them to settle, with rafts or whatever, but as oysters do. It's just because you've got selection all the way through the larval stage and then all the way through the growth stage. I'm just wondering how you...
So you do indeed. So the life cycle I showed you is a single generation life cycle. And I think of the larval part of that life history as this impenetrable black box, because it's very difficult to know what's happening there in terms of selection, exactly where they're going. You know, we're getting small inroads into ways of looking at that. Certainly there's going to be a lot of selection happening there, much of it in terms of the environmental gradients that I've been talking about. Some of it's going to be stochastic, some of it's going to be predation. It's a huge amount of mortality. So I don't know how much of the patterns I'm seeing is happening in the larval stage and then settling down, or versus the larvae being homogenized, settling down, and all of it being post-settlement selection. You're exactly right that I can't distinguish those two. But I think it's sort of helpful to think of it in terms of post-settlement selection, because that's going to, well, it's just a simpler way of presenting it, I guess. We don't know yet. Yes?
Hang on one second.
Kind of a counterbalancing factor would be diseases. We know the oysters in the more saline areas are more susceptible to some of the diseases that cause high mortality, and the opposite is the condition in low salinity conditions. So the low salinity may have a certain, bad effect. On the other hand, they're not affected by some of these diseases. So you got those two balancing factors that may, counterbalance and may cause some of that differences you're seeing in population. Is that a possibility?
The question is about the fact that disease pressure also varies along the salinity gradient, with two major pathogens having much stronger effects down near oceanic salinities and having much less disease pressure up at low salinity. And I didn't even mention disease pressure, but I think it's very interesting how these two patterns of stress, salinity and disease, might interact. And I wouldn't be surprised if some of the genetics underlying response to salinity gradients and response to disease is maybe entwined. So that's, I think, a big research question about understanding the dynamics of the evolution of disease resistance, and whether there are constraints on that, because the genetic architecture involved in dealing with salinity is also involved in disease resistance, and therefore you can't do both very well. So I want to come over here and start at the back. Pete?
Couple questions.
See how far we can, close we can get to.
If the low salinity pressure in the upper Hudson is sort of early in the spring, and the oysters are spawning later in the spring, then wouldn't you expect that selection pressure to happen after their first year? And if that was the case, wouldn't we see a lot of dead one-year-old oysters as opposed to oysters dying before they settle?
Sounds reasonable.
That's one question. And then the other comment is that if the reduction in genetic diversity is the same after one generation, the Soundview example at Cornell, as it is after 20 generations with the Fisher's Island example, wouldn't that's, I mean, if it's strictly genetic diversity, doesn't that makes me think that it doesn't matter where you're getting your oysters from, because that reduction is something that happens once. And then my question that I've been wondering for a long time is, can you model how quickly those oysters, if they were restored, would return to that original genetic diversity or some comparable genetic diversity once they've had a few opportunities to spawn in the wild? And then my only other comment is that those Fisher's Island oysters, which are from a high salinity hatchery, there's two different nursery sites. That's my parents' oyster farm where I grew up. There's two different nursery sites where they have upwellers. One is in a salt pond where the salinity averages from 10 to 20, and the other is in the Pawcatuck River, which separates Connecticut and Rhode Island, and the salinity there is anywhere from 0 to 10 in the spring. And the oysters grow best in the Pawcatuck River once there's some salinity, some salt in the water, but often that's only 5 to 7 parts per thousand. And those oysters grow fastest in that river. I mean, presumably because of the food that's present there, but also, I mean, that's a high salinity oysters. They're originally flowers, 20 generations at Fisher's Island at 35 parts per thousand, and they grow really well at the Pawcatuck River.
I know, and you guys, your dad is sending out oyster seed to a lot of different conditions, and it's been doing that. So I'm not trying to say that Fisher's Island oysters, as a function of this genetic bottleneck, are somehow deficient. I'm saying we need to do some tests to see what are the estuarine conditions under which that kind of a bottleneck matters. And really, it's not just that these oysters coming out of a hatchery have lower genetic diversity. What's also happening in a hatchery is domestication selection. It's very, just like I've described in the estuary, there's very strong selection that can happen in a single generation. It's going to happen in the hatchery as well. Okay, in the culture in a hatchery, as the larvae age, there's cleaning of tanks and filtering the larvae, and you filter out the small runts. Those are the slow growers. And so even if you're not trying to select for fast growth, you end up inadvertently, to keep a clean culture, selecting for fast growth. Okay, so so if you've done that for 20 generations versus one generation, they might show the same level of overall genetic diversity, but the functional genetics relating to growth rate and some other traits, who knows what, may be having an influence in terms of their performance. So we need to do the test at the phenotypic level. Yes?
general question. What is the population of the wild oysters in this area? And what do you think it'll be in 5 years?
Well, that's the $1 million question, isn't it?
No, they're out there.
So the question is, how many oysters are out there, and how many are going to be out there in 5 years? Well, there's going to be 1 billion in 5 years, I can tell you that, at least. What's the time horizon for the Billion Oyster Project? Right now, we don't know. We only just discovered a couple years ago that there's living oysters, really very vibrant oysters up at Tappan Zee, right? How many other subtidal reefs of oysters do we have around? We just have had very little survey work. So nobody can give you a decent estimate of how many oysters, but it's a remnant population.
I mean, people have been, I don't know, looking into it for quite a few years.
Not very extensively, very locally. So you really, in order to understand the population in the estuary, you need to study at the estuary scale and study across the different habitats. And most of the research has worked at the shallow subtidal or intertidal because that's where we can work effectively for relatively little money. But we now know that subtidal oysters are maybe one of the ways that there's a remnant stock that's doing well. So I think that's an important question. The amount of recruitment that we've seen, the amount of spat fall that we see in a good year, that basically means an environmentally fruitful year for larval survivorship. We see quite a bit of larvae ending up as spat around the East River and in parts of the Hudson and up in Hastings. That tells me that there's a pretty sizable remnant population. Where they are and how many there are,
You think they're hiding?
I don't know. They're hiding. They're hiding from us so far.
When you were doing this study,
when you were doing this study, did you run into any complications such as disease, Demodex?
Thanks for the question. Did we run into any issues with disease in these oysters? We did not have the funding to test for disease in these oysters, so I can't tell you how many of the oysters that we've analyzed had infections from dermo or MSX or anything else. Unfortunately, it would have been nice to be able to collect that phenotypic data and try to relate it to the genomic variation. Maybe next time, because those are important questions.
Similarly, did you do any other water quality testing in the sites that you were taking oysters from and monitoring?
We had sondes collecting salinity, dissolved oxygen, temperature data at some of the sites, and we put other sites near where there was other water quality monitoring. So we have those basic data for most of our sites.
And pollution too?
But no pollution work, no. Again, that would be, especially when we're collecting genomic data, it would be great to be able to have much better environmental data and really good physiological phenotypic data to relate to the genomic data. Working your way around again?
in terms of whether it's the larval stage or after settlement where the bottleneck is on selection, it would be possible, wouldn't it, to be able to do the genetic work on spat? So if you could compare the genetic diversity in a given area of the spat versus the adults, you could see how much selection was going on post-settlement.
Yes, I completely agree.
I mean, if they can get enough from a Neanderthal, they certainly can get enough from
Yeah, so that was part of the experiment. So the question was about comparing very young spat that are presumably closely representative of the genetics of the larval pool versus the adults, so the post-selection genetics. And our sampling design, some of what we did was an attempt to look at that. We've got both spat and adults from both Hastings and Soundview, And in addition, we took the hatchery-produced oysters from the Southold hatchery, put those out in cages at Soundview and Hastings. Over the summer, over about 3 to 4 months, didn't see a whole lot of mortality. I mean, like 30–40%, but not enough that that I'm seeing a strong genetic signature of those effects yet. So I completely agree. It's a one experimental approach, making that comparison before and after the presumed selection occurs. That we're trying to approach it that way to see to get that before and after picture.
Just like to say that, Lamont already did the side scan sonar up the river, and that was about 12 years ago. And that was the first time I heard about the beds up at the Tappan Zee Bridge. So it might be interesting to go through that data and see, if they documented any other wasted beds in their trip up the Hudson, and that might give a clue to where this, recruitment is happening.
So we look at that quite a bit for the work that Matt mentioned going on at Tappan Zee, and it actually looks like there are not a whole lot of pockets that look similar to this small pocket where we found the relatively high density oysters. We're going to do a little more of that work this summer, but Judy mentioned that there seems to be not a lot of work below Tappan Zee before you get to like Pier 40, but there's also not a lot of evidence that there are oysters there. We don't understand exactly why.
Right. Well, were there any other pockets at all?
There are some reflections that look somewhat similar, but we've done only very few grab samples to look for that. But the few that we have looked at have not found the high densities that we've seen at this one little site up near Tappan Zee.
Pete?
Just like to the where are the oysters and how many oysters question. The amount of the subtitle New York Harbor or Hudson River Estuary where we actually look is extremely small. So there's something you can infer from like side scan data and stuff, but where there's actually been grabs, it's much less than 1%. So there could be like we have not done the survey work to say that there are or not oysters in the vast majority.
And it's going to be a tough.
No, there's just people have not looked. So even all of the recent energy that's gone into looking, most of that is intertidal, it's not subtidal. There's you have to be a scuba diver or have to do grab samples. It's a real challenge. There's very little of that. Scuba diving, Chris is not a scuba diver. You can snorkel.
So.
Yeah. Well, anyway, talk to you later.
Someone needs to invent a custom carbonate measuring tool so they can just like float along the surface and calculate these things.
Liz, you got a question?
Yeah. So for both the RNA-seq data and the DD-RAD data, were you able to, or did you attempt to map any of those loci to the Pacific oyster genome? I'm thinking particularly for the RAD-seq data, it would be very interesting to know if those loci that are dropping out from the hatcheries, that diversity that's dropping out, if that's specific in any way, or if you're just looking at a generalized bottleneck.
Yeah, so that's work that's ongoing. The question is about whether we've used the Pacific oyster genome, the full genome sequence is a wonderful reference that we can make use of, mostly with respect to coding sequences, because they tend to be more evolutionarily conserved. Everything outside of coding sequences the sequence is just too different to be able to use that Pacific oyster reference. So the work in my lab is ongoing in terms of, with these data, testing for genetic, for polymorphisms that are showing patterns inconsistent with neutral genetic drift and more consistent with selection. So for example, we're using our environmental data, our salinity data, to test for correlation between genetic variant frequencies, the frequencies of a genetic variant at each of our sample sites. Is that correlated with the mean or salinity at those sites, or the minimum salinity that was seen over the year? And in some cases, we're seeing some hits, and those, of course, we're trying to find some functional information. What does this gene sequence do? But that work is still preliminary enough. I didn't feel comfortable presenting it.
Maybe time for one more?
In the first test you did into Delaware, the plasticity versus genetics and the two different salinity, maybe you had a slide and you didn't see it or understand it, but just mortality, were you seeing significant differences when you took the high salinity oysters and put them in low salinity or vice versa?
Can you repeat the question?
Yeah, so the question was, did I see any difference, did Laura, my student, see any difference when she took low salinity oysters from Delaware Bay and put them at high salinity tank, or the high salinity oysters at low salinity tank? Indeed, she did find the only, there was sort of background level mortality of all the oysters, it's like less than 5%. But the low salinity oysters did not like being at the high salinity. So those we saw more like overall 30% mortality. They seemed less resilient in that sense. And that's interesting. It corresponds with our gene expression data, in which I didn't really show this, but but in terms of those reaction norms, in terms of the response of gene expression to different salinity treatments, we saw the oysters from the high salinity reef showing a big response in gene expression, so a lot of plasticity, relative to the oysters from the low salinity reef showed kind of a weak plastic response in gene expression, which again you might think relates to resilience. So thanks, that was a good question.
And last question, just exactly on that line, and you certainly think that there's a selection for this osmotic regulatory capacity somehow or other. I mean, the great thing about it, I think, from your perspective, is without having to go multi-generational, which we never do in ecology, you could look, because osmoregulation has an energy cost, you could look at the energetics to support osmoregulation from low to high. So basically, there should be a genetic marker in there someplace that you can, where you don't have to just do it kind of whole cloth, but you can partition it into actually what the cost is for that 6 part per thousand oyster versus against the 30.
Yeah, so the question is about relating the osmoregulatory function that we're looking at to the energetics of the oyster, and I completely agree. I mean, you've got to ask yourself, why are there thousands of genes responding to this salinity gradient? Does it really take that many genes to control cell volume? Well, it is a complex phenotype, but what we're seeing there probably is a whole shift in the metabolism of the oyster, dealing with these different patterns of stress that they see. And so I completely agree. A fuller energetics and metabolic approach would be is an important way to go. Thank you very much for all your